Hello, today I'm going to talk about radiobiology research and the progress and why they matter for space exploration. My name is Francis Cuccinata. I'm at the University of Nevada, Las Vegas. I want to acknowledge Tom Hay and Leonard Fisk for sending me the invitation for this talk and my funding by the National Cancer Institute and the University of Nevada, Las Vegas, and many collaborators. I mentioned a few here on this slide. So I'm going to talk about some background on radiation protection, why environments and shielding properties are fairly well known, and why radiobiology research, the health risk, are the most important areas to be addressed. So as an introduction, because space missions are currently funded by government agencies, there are going to be requirements for acceptable risk levels. And our argument is that the uncertainties in the projection model are still the major issue. The risk may be higher or lower than the conventional models to project risk, but because the uncertainties are so large, they're indeterminate how well we're predicting the risk, whether we're above or below what would be acceptable. And this also feeds into countermeasures. Do you need countermeasures? How would you design a countermeasure if you did need it? It's the same uncertainties have to be addressed. And then these are the major issues, not environments or shielding. I'll talk a little bit about the background there. So space safety requirements in the US, they have the National Research Council and the National Council on Radiation Protection Measurements that have provided the guidance. They use a 1%, 1 in 33 or 3% limit because of the uncertainties in high LET radiation. We also have an ancillary requirement of a 95% confidence level. Then during the mission, you would want to prevent radiation sickness, cognitive detriments that would affect the mission performance and success. And then you also would want to limit other late effects like central nervous system and circulatory disease risks. All this feeds into the mission and vehicle requirements, shielding, dosimetry, what countermeasures are needed, what crew are eligible for a specific mission, how long can the mission go, and then finally, in the past, you've always followed the ALAR principle. Maybe perhaps for exploration, it would be different, but that would be important consideration for past missions in low Earth orbit. The environment, so we have the galactic cosmic rays. This is kind of familiar, but I'll mention it. Here, the energies are very high. They originate from supernova explosions, is the main theory, and then accelerated in space before they reach our solar system. Because of the energy not effective shielding, and then the solar particle events. So the cosmic rays are a steady background varying about a factor of 2 over the solar cycle. And the solar particle events are more infrequent. They vary from cycle to cycle of 11 years. The last decade has been very low frequency of solar particle events, but in other cycles, they've been quite high. But unfortunately here, shielding is effective. So you can optimize the shielding materials to reduce the weight and the configuration of the shielding, but the main thing is operational event alert and responses. Weather or forecasting can help, but usually would do a mission without that capability. And then the issue returns then passive shielding, but with alert systems. Cosmic rays, I remind you, they're heavy ions, and the median energy is 1.5 GeV per nucleon or 1500 MeV per nucleon. So they're not easily shielded. And then they also interact with the spacecraft and tissues to produce secondary radiation, including neutron and low energy, high LET recoils. So give you a little bit of background dose. So dose is used loosely, but absorbed dose is the amount of energy deposited in bulk material or per unit mass. Units, joules per kilogram is 1 Gray. The older unit was 1 rad, and then the fluence is the number of particles per unit area. That's can be estimated as the dose can be estimated as the fluence times the LET, the linear energy transfer. So it's equating energy loss with energy deposited. And then the quality factor is introduced as a sort of like a fudge factor to account for the differences, types of radiation. But that's where we want to address a lot of our talk today, is the basis for that and whether that's really a good quantity and a good approach. And the units of the dose equivalent or effective dose are sieverts, which is the absorbed dose in tissue times the quality factor. Okay, so just give you more hints on dose. So this is a picture of a Monte Carlo simulation of protons interacting with a volume of water. So water makes up about 70% of tissue mass. And what is shown is the position of ionizations and excitations. This has been normalized in a 5 by 5 micron box to 1 gray. So this would, if you take an LET of a high energy proton, 0.3 keV per micron, that corresponds to 450 protons. And then if you start then saying, well, what's the dose? In different regions, we can average the ionizations and excitation over different size voxels. So going from 20 to 40, you can still see there's black areas where there's no dose and other areas that are hot spots much greater than 1 gray. And then as you make the voxel size much larger, you can see it becomes more uniform and steadily approaches that 1 gray uniformly through the box. It's still a very small volume or order of the volume of a cell nucleus. And remind you that a DNA nucleosome is 10 by 5 nanometers. So this heterogeneity affects the energy deposited, the number of ionizations of a DNA molecule. And then other biomolecules are usually on the order of a few tens of nanometers. So if we go now to a high LET, so this would be a iron particle of 1,000 MeV per atomic mass unit, LET 150 keV per micron. And now we have just one particle makes 1 gray, but you can see that it's very non-uniform. So even if you start averaging these voxels over larger volumes, it's still many areas that are black, meaning no ionization, no dose. And then you have this core part where the dose is very high. So in a very small volume, the size of a DNA molecule, you're getting 10 to the 5th gray. But in other areas, you're getting 0. So the averaging takes a much larger volume. So very different. And we say that this leads to a qualitative difference. We want to know, is that qualitative difference based on the physics and early chemistry, which is fairly well known, does it affect the health risk from these particles? So the environments are well known in the sense that we have such a good feel of the past, and we can represent data to such an accurate extent that we can say we have an environment that we can design to. This is some examples for GCR protons and helium versus the Voyager 1 data. So that's interstellar spectrum, and then different times in the solar cycle. And this is for iron, neon, and silicon. This is a model I published in Life Sciences and Space Research about a year ago. And this just shows you the solar modulation. So we certainly want to design to a reasonable worst case, so solar minimum. Point out that at solar minimum, even though one solar minimum varies to the next, the variation is at lower energy. So you usually typically don't see it for normal spacecraft shielding thicknesses or within tissue because the low energy part is shielded out from the tissue. About solar particle events, so this shows you historically 30, 60, and 100 MeV protons, their size, how many protons per unit area per centimeter squared. Point out for tissues, this higher energy, 100 MeV, is the most important one. So it's much less frequent to see anything. The largest is approaching 10 to the 9th protons per centimeter squared, but none actually make 10 to the 9th. So that would be the rule of thumb, is to say, what's the probability to get these large events in a certain time period? And we've done that in some of our publications with Young-Hee Kim and Albert Fivison a few years back. This just shows you the doses. So this is the largest 43 for light shielding. This is the effective dose. So it's average over the organs. And you can see that some of them do exceed dose limits and perhaps get into the region of acute radiation sickness, maybe a half dozen of the largest 43, but with reasonable amount of shielding. So 20 grams, that's like the average shielding on the space station. You knock that probability down to almost less than 5%, and usually you'll have a storm shelter, and they should design an exploration vehicle with a storm shelter approaching about 40 grams, and then you would have a 0 probability to approach acute radiation sickness, and you might at most have a residual cancer risk. So definitely the cosmic rays are the main issue. To describe the cosmic rays, you need to know the nuclear cross-section. We did a lot of work on that, and they're not perfect, but they have we have a lot of good models, and they're fairly accurate. This is the fragmentation cross sections. Iron is an example, but similar for other ones. Pions are made in secondary reactions. There's a model we published showing the inelastic and total cross section for pions with aluminum. And this is a helium double differential cross section in the longitudinal and transverse directions. So we have a good feel for the cross sections. We can use those for transport codes. This is Bragg peaks. So the depth dose distributions for different heavy ions measured at Brookhaven compared to the model. The model tells you how much is from secondary. So this is fairly well understood. We can use those models and make predictions in space. And you can see that, again, they're within, for integral quantities like dose and dose equivalent, they're within about 10%. There's some other areas about details of the spectrum, but it's kind of a little bit forgiven because if you mix the spectrum in one area and it's got to be compensated in another area, and since the quality factors at high energy are changing very slowly, it's not much of an impact of that. There's been a lot of excitement always about space measurements, but I just want to point out that so we had a paper a long time ago in 2003, and if you look at the dose rates we reported for different Apollo missions, shuttle missions, NASA Mir missions, they agree very well with these. So I say these measurements validate what was known, and they're not giving us tremendously important new information. So we can use all that to say shielding is not a big factor. We know that here, for example, polyethylene aluminum provides some protection at solar minimum. This is the loss of the low energy particles, but you're continuing to lose higher energy particles, but you're making new particles for secondary, so you get a flat dose response. And at solar maximum, where the lower energy particles are missing, it's a very flat response. You certainly will get a bigger decrease as you approach 2 meters, but that's impractical for space vehicles. So the focus then is on the risks. These are the major players. Cancer, emerging over the last 10 years is the central nervous system cognitive risk. Acute radiation risk, I argue that you can protect yourself operationally. There's not a big radiobiology question there. So the main issue is the cancer and cognitive risk and whether there's a qualitative difference. We know there's a quantitative difference, but is there a qualitative difference because of the energy deposition and that early chemistry that would occur? So we certainly have evidence of that for cataracts. So there's a paper we published a long time ago in 2001. This was followed up by state-of-the-art measurements by Leo Shilak. So what we find here is these are doses, average dose of 8 millisieverts. So that's much less than a Mars mission. And what we did was we grow, so the high dose has an average of 8 millisieverts and the low dose below that. And you can see there's a separation between the two population of Apollo, Gemini, shuttle, NASA Mir, and Skylab astronauts. And this non-trace cataracts is one that has some evidence of visual impairment. So you see a separation of the two groups at a very low dose, and you certainly see a risk within about 5 years. So this becomes a problem because first, we don't expect this from X-ray. X-rays, you would have a threshold that's observed closer to 1 or 2 gray. So the lens dose limit is being violated. That to me is a minor concern because if you return from space and have a increased risk of cataracts, it's easily corrected. But the question becomes then for a Mars mission, would it occur during the mission at the higher doses, like 100 to a few hundred times higher dose? Would this occur within a year or two? How about other late effects? So we're wrong here. X-ray doses would not predict this. X-ray effects in humans and animal studies do not predict this. So it's a heavy ion effect. And we have strong evidence of that in our papers. So it's an aging effect to the lens. Does it affect other tissues? And point out this is something that's a violation of Einstein's theory of special relativity as applied to biology. So he suggested that twins, one of them going on a long space voyage would come back younger, but we're saying they'll come back older because of the cosmic ray exposures. So CNS risks are concerned as early and late. And first point out, it's a non-stochastic risk. So later on, I'll talk about cancer risk, which is stochastic. This means we're taught that there's a dose threshold. It's a dose threshold that we don't know. People have not spent any real effort in understanding where the threshold is. And also for deterministic non-stochastic effects, we expect the severity of the effect increases with the dose above as you pass the threshold. So unless you do the studies at low chronic exposures, we don't know what's really occurring in a mouse or a rat. And then there's the issue about going to humans. So we know the doses, and we have evidence at higher doses that some risk occurs in small animals, but we don't know for a low dose, like a Mars mission, and a chronic exposure, and what would happen in humans. So this is definitely an area where a lot of research effort is needed. So the issues that have been seen are the reduction in the number of neurons at higher doses, but how about at a lower dose? Persistent oxidative stress, so that's also a concern. So what we know from the past is based on largely cancer patients with brain cancers that are treated with radiation. They found certain types of injury. Early studies in space radiobiology suggest that those types of injuries don't occur at the lower doses, say below 1 gray of heavy ions. So we're seeing more subtle effects. So the question is, are we going to see these subtle effects that would affect cognition during the mission, and would they increase the risk of late effects? So the main approach that's being used is a study in mice and rats, effects of different types of particles, different doses, with a range of cognitive tests. To remind you that the brain has different areas, and different tests of cognition can test certain areas. One area that's similar in different species, mammalian species, and even others, is the hippocampus. So that's a good one because we know more about it, how it would translate to humans. The other areas, like the prefrontal cortex, it's more elusive. We know that humans have a much more developed cortex than mice and rats. So how you translate from studies that cognitive tests that study in those areas to humans is less well understood. So to give an example that we studied in some recent papers based on a meta-analysis of different data is the novel object recognition. This is from Bernie Rabin at University of Maryland. So what they do is they train the animals to find an object, and then they introduce a novel object that they have not been trained, and then they measure the time it takes for them to explore the different objects. And this involves the medial temporal lobe, the perennial cortex, and with certain roles for the hippocampus and the prefrontal cortex. So here's an example. So this is from Bernie Rabin again. So 600 MeV and 1,000 MeV iron particles in Sprague-Dawley rats. Here they see nothing at 10 rads. So 10 rads is above the Mars mission cumulative heavy ion dose, all the heavy ions, even the lower Z, lower LET ones. So this would suggest there's no risk for astronauts. Again, many question marks. Here are 50 rads, but they didn't test between 0 and 50. So what we did is, in a meta-analysis, we introduced a familiar to novel object recognition ratio based on the reported times of the different investigators. And then we introduced a relative risk model of that. And we then replot all the data from different investigators, over 60 experiments, 60 publications. And you can see here's where we're interested in. It's not clear, but we went ahead to assume there's a linear response here, tested up different models of how to represent the radiation quality, and then made some predictions. So for the average solar cycle, the excess risk is less than, say, 1.1 for a Mars mission. So, and then with the uncertainty, we would certainly can't rule out that there's no risk. And that's for a mouse or a rat. Now this model, NOR, is also used in studies of different disease, Alzheimer's disease, Parkinson's disease, traumatic brain injury. And so we can then make comparison of the relative risk from reported observations in models of these diseases with the Mars mission prediction. And you can see we're quite a bit lower than most of these. But the TBI affects their vision, so that could also impact how well they recognize an object. And Parkinson's disease has different aspects. So we're saying that they're potentially a risk, but it's not really conclusive based on what's the evidence so far from experiments. Another one is spatial memory tests. This one we found that there probably is a threshold and nothing is going down. It's a single investigator might have seen an effect, but if you look at a meta-analysis, it suggests that there's probably no effect for a Mars mission. Mind you, this is the doses from different charge groups. So most of them have a dose less than 0.1 rad cumulative dose, even though the sum of all of them might be as high as 5 rads, or 0.05 gray, exception protons and helium. In a lot of the cases, they don't see an effect with protons and helium, which are low LET radiation. To turn to the mechanisms, so this would be important to establish how you would translate the information from animals to humans. Also, maybe give you some insights on dose thresholds, what's the cause of it. If you know the cause, and if it is a concern or a bona fide risk for a Mars mission, an approach to a countermeasure. So the support cells to neuron cells, the vascular and the glial cells, neurogenesis is being impaired at higher doses. The structures of the neurons, which we call neuron morphology, is being altered. And then there's some evidence of epigenetic changes, and we're going across most of these as neuroinflammation. So we modeled the neurogenesis. So this is one that we're going back about 10, 15 years. There was a lot of studies with heavy ions and interesting results at half gray or higher, but at lower doses, we weren't sure. So we modeled this with a mathematical approach, a nonlinear math model. The key thing is the neuroblasts, which arise from the neuronal stem cells and the mature neurons, and also the glioblasts that arise from the same stem cell. These two are being killed off at higher doses, but they're also sending signals back to impair the proliferation of the neuronal stem cells. And all those things can affect cognition. So we modeled that, and then different data sets to show you. So, the doses are higher in different time points, different types of radiation. There is an inflammatory role at higher doses, certainly at 1 gray, there's increased change in impaired inflammation. There's also evidence at higher doses that the stem cells are switching from the ratio of how many neurons are made versus glial cells. But again, at lower doses, we're not sure what's going on there. So we made some predictions with our nonlinear kinetics model, and we see that most of these radiations would not be a concern for a Mars mission dose. Iron particles, you can see there's some transient changes at 0.1 gray, but it doesn't seem to be too important. If you went lower, where we would be concerned, it doesn't seem to be a big issue, and then we could predict some threshold dose. So this suggests, and John Fike, who did some of these experiments originally, suggests the same thing, that impaired neurogenesis is not an issue for a Mars mission. Now, the other part of this is genetics. So most of the studies with heavy ions on impaired neurogenesis are with the C-57 black 6 mouse models. We did a paper that was published in the Frontiers in Neuroscience journal that looked at in rats with X-rays. So we don't have this information for protons or heavy ions, but they showed with X-rays that there's many different responses for different strains of rats. Again, that becomes a big unknown for heavy ions. Would you see the same range of genetic differences? Which genetic strain would represent the average astronaut? The mouse hippocampus has been studied. This is just a picture schematic. You have the axons and different neurons, and then the neuron has the soma and the dendritic tree. So they use tree jargon, and then on the tree you have the different branches, and on the branches you have the spines, and they make the synapses with the axon. So that's where your memory is stored, and new memory, spatial memory, things like that are controlled in the hippocampus. So the UCI, University of California Irvine group, has a few publications on this where they use a transgenic mouse model that has a fluorescent reporter for the spines and the dendrites, and then they can measure the changes in those. This just shows you at fairly low doses, acute doses, they see these changes. And so it's potentially important. Again, this is a single mouse strain, and this different types of radiation, not a expansive set of types of radiation, acute, not chronic. So we simulated this with the Monte Carlo models. We have full stochastic radiation tracks passing through the hippocampus and passing through individual neuron structures, and come up with a model for the degradation. So we have a model that says the loss of the cell is due to heavily damage to the soma, but also if these branches are trimmed too much, then also trigger apoptosis. And that's known from fruit flies and C. elegans that would occur, less known in mouse. But we expect that if too many of these branches or too much is lost, then that would lead to the loss of the neuron. And then we can make predictions. So there's an example for titanium, heavy ions, and dendritic granular cell neurons. And one of the things we wanted to point out is that there's going to be a difference in how well, in the information, whether you count the damage on a single neuron or in a population. So this is simply because in the population, they don't have a way to count how many cells were lost. So they're looking, say, 30 days after the exposure, they're looking at a plane and they don't have information because it's very complicated, the clustering of the different neurons. How many neurons were there originally and how many neurons are there now? We're just counting the branch length or the spine number in the whole population, but the population hasn't been normalized. So we made an estimate of how to normalize it, and we say that you would get this divergence of results, especially at higher doses, because you don't have that information. So the summary of the CNS risks I'll make before I turn to the next part of my talk is that I still don't think the risk is established. So I don't think countermeasure research is the right approach at this point. You might have a countermeasure that works because there's no risk, but you don't have to establish the risk. As far as I know, I know from being involved in the field for many years that there's underreporting of negative results. I think there needs to be emphasis on that. There's not enough information and studies on threshold doses. If they see the effect at 0.1 Gray or 10 rads, they should go down to 1 Gray or 0.01 rads. 1 rad or 0.01 Gray, or even lower. It was important from space station, it's even lower. Where is the threshold if the risk is true? And then how do you go from mouse to humans? We saw that there's a lot of genetic effects. And then another one is combined stressors, that you have this microgravity that changes inflammation, immune responses, sleep deprivation, interaction with pharmaceuticals used by crew. All these things need to be considered. So again, too early for countermeasure studies. More information on is the risk really occurring at the right doses and a chronic exposure is needed. How about cancer risk? So the rest of my talk, I'll focus on this. So cancer risk is the one that we know it exceeds the dose limits for a Mars mission for most of the scenarios. The other ones, it's emerging information. So we can use what I'll call the conventional model and apply it and say that the risk we're going to probably exceed 3%, and the 95% confidence interval will be even well above that. Now, the traditional or conventional model is loosely based on the idea that radiation is an initiator. It causes DNA mutations, chromosome rearrangements, and a series of events occurs due to those mutations that lead to the development of a tumor. However, there's a lot of research now that shows that tissues controls and inflammation have a big role. So this is a schematic from a paper by Mary Helen Barcelohoff at UCSF in Nature Reviews Cancer. And she has a very interesting and informative model where it's a chimera model of a mouse where she removes the epithelial cells in the mammary gland and she irradiates the fat pad. And then she puts the normal epithelial cells back and she sees tumor growth. And some more recent papers from her show that it changes the infiltration of immune cells that normally would respond to damaged cells or mutated cells at early stages. So this plays a big role in cancer risk. So we need to think in terms of all these things together, not just that radiation is causing DNA mutations, but also it's causing changes to tissue microenvironment and changes to how the immune system responds to damaged cells. So the model we developed about 7 or 8 years ago is called NSCR. And I continue to revise it. The original model was reviewed by the National Research Council and then the NCRP, and then a more recent one in 2015 by the NASA External Review Board called RCAP. This model integrates all the low and high acuity model data for chromosome aberrations, mutation, mouse tumors, and to make a new version of the quality factor with the uncertainty distributions, which is different than the ICRP or the NCRP approach, based on track structure concepts. We also look at going from epidemiology data of incidence and then converting that to mortality rather than using historical mortality data because of the changing cure rates and cure rates as a function of different host nation that would affect your prediction. We also use never smoker models for astronauts rather than something like the average US population. So this is a prediction from one of the earlier versions. You can see this is the risk of cancer incidence for different types of tumors, and then heart disease, and then the total cancer risk and the total risk of cancer death, so incidence and death. And you can see that the risk of death for cancer alone could be higher than 88% for females at the 95% confidence level. The PC is the probability of causation here. I estimate at 10 years post-exposure. So this is a PR nightmare. If you do survive the mission and 10 years later you're getting cancer, what's the probability it was caused by your Mars mission exposure? So I say it's a PR problem because these programs seem to be spread out over decades. So early on, you might see something like this occurring, and that would be problematic for continuation of the program. If crew are coming back with leukemias or stomach cancers, you have a very high probability it was caused by either radiation exposure. This would be a prediction for males. So again, a little bit lower because of the differences in lung cancer between females seem to have higher, but also breast and ovarian cancer. So the uncertainty is driven mostly by the quality factor. So the quality factor parameter that I didn't have much time to discuss is like this parameter here, that's the strength. And this is all the mouse tumor data. You can see it varies almost a factor of 2 orders of magnitude. I took out here liver because they seem to have the highest RBE in male mice, and the Hardian gland because this is a model that's been used a lot, but it doesn't appear in humans. And so this brings the distribution much narrower, but it's still more than the order of magnitude. So that's a problem. What is causing all that uncertainty? Along with this is the qualitative differences in cancer risk. So the quality factor approach of the conventional model only takes into account quantitative differences. So we're very concerned about it. Is there evidence for qualitative differences? So one evidence is the shape of the response. This is X-rays and gamma rays and protons as a linear quadratic shape. High LET heavy ions and older studies with low energy neutrons show this very dramatic rise and then a flattening. What else? The tumor latency, the high LET radiation appears to have a much shorter time from irradiation to time of tumor. So that's a concern. And then the spectrum of tumor seems to be different. The mean life loss is the bottom line for the 3% cancer risk. So we can think of a conditional probability. Are you above or above the limit, say 3% or 5%, whatever you like. But the risk is how much life did you lose? So it's based on fatality, which I think is a correct way to do it. And you can see for leukemia, for a 40-year astronaut, they would lose 23%, 23 years. So this is, if they got leukemia, they would have lost 23 years of life on average, distribution around that. Most of the solid cancers are around 15%. There's also concern about heart disease. They were about 9 years of life loss. A little bit different for males and females. Females live longer, so they would give up a little bit longer life. What else about quantitative differences? This is from Bob Aldrock, older study at UTMB. He's showing the rates of metastases are different. So gamma rays and protons are similar to background liver tumors, but heavy ions are more metastatic at a rate of about 3 or 5, fourfold higher. And then Al Fornes at Georgetown University in a transgenic model of colorectal and intestinal tumors show that the heavy ion ones are very invasive and more malignant, and gamma rays are protons, which are, again, similar to the background tumors. So this would affect your uncertainty distribution. This is the model with estimates of increased lethality of the tumor. So you can read our paper how we estimated that, but it drives the uncertainty and the risk prediction much higher. How about the non-targeted effects? So this is really the main one that more important uncertainty, and one of the main ones I think where new knowledge and new evidence, new experiments are needed. So what do we mean by non-targeted effects? So we think of a radiation hits a cell, DNA damage, misrepair, that gives you mutations. Non-targeted effects models say that the irradiated cell isn't the one where the risk emerges, but it's the signaling from the damaged cells to the neighboring cells. So that's a bystander effect. Genomic instability talks about descendants of the irradiation cell in a tissue having leading to the risk, and then also the microenvironment changes. Abscopal effects are for partial body irradiation, where you can see the effect in a tissue was not irradiated, caused by damaged immune cells or change in signaling from the immune cells as they go from damaged to unirradiated tissue. So there's different ways to look at this. In the interest of time, I'll focus on the physics way. So this would be, if you irradiate a cell to a certain dose, you know on average how many cells were hit by the radiation. So a nice experiment by Ed Azam and Proceeding of National Academy of Science, he used a very low dose, 0.003 Gray. And at that dose, you know that only 1% of the cells are being hit. He studied this marker P21, which is a DNA damage response marker related to cell cycle controls. And he sees that this marker is increased in more than 10% of the cells. So you would expect if it was increased only in the DNA, in the cells directly traversed by the alpha particle, only 1%. Also, he used an inhibitor of gap junction and intercellular communication, and you can show that that takes the effect away. Other people have looked at also reactive oxidative scavengers and shown the effect. The same thing, that if you apply those, the effect is reduced. Another way to look at this is to look at the dose response. This is for neoplastic transformation in a cell culture model with high LET alpha particles. So this is the normal way you have dose transformation frequency on the y-axis. I take this data and I divide the y-axis by the x-axis, I get the frequency per dose. And if it was linear, you would have a straight line. So when you then it changes to fluence rather than dose, you can see at higher doses, it's a linear curve, or excuse me, a constant curve. Right? A linear divided by the dose would be constant, but at lower doses, less than 1 per cell, you get this rapid increase. And this is the dose area we're interested in. We're not interested in this area. And then this would be something from my past studies with Megumi Hara and Carrie George showing the same thing for chromosome aberrations. You divide the y-axis by the dose. It should be flat if it's a linear response, but it increases as you get below 1 TREC or 1 particle per cell. This leads to a 2 to 3-fold increase in the cancer risk prediction. And it shows you for the, before we had the upper limit 8 or 9% for males and females. Now we have it more uncertain, but possibly approaching 20% for females and 15% for males. So this, what was once higher than an acceptable limit for space station, maybe backed off for a Mars mission, becomes problematic if it becomes more like Russian roulette, higher than 15%. So the other issue is about the synergy or the combined challenges of microgravity, radiation, sleep deprivation. So this illustrates in neuron cells what the problem would be, is as you have a prolonged dosing period, you're also prolonging the activation of neuroinflammation. In this way, in this curve, they're talking about microglial activation, which is a surrogate for neuroinflammation in some sense. So at some point, you're going to exceed the onset of disease. So if you have a threshold, the combine of the damage and the chronic inflammation could exceed the response. So increase your cancer risk, take you over the threshold for non-cancer risks like cognition or heart disease. So on the space station, when they return from the mission, during the mission, they have some evidence of changes in immune responses and inflammation. They seem to also go back to normal post-mission. But for an exposure on the Mars mission, which is a dose like 5 to 8 times higher than a space station mission, and a much longer time period, we have this inflammatory response from the combined challenges, and it becomes a problem. So summary then would be the conventional model says at the upper 95% confidence, you're less than 10%. So maybe higher than what's acceptable for in the US program, but perhaps you could accept that for an exploratory mission. But in these models that look at qualitative differences between high and low AT, high being mostly the heavy ions and some neutron contributions, you could approach 20% with higher life loss prediction than the conventional model. And then combine that with altered immune responses. So we think these are the major areas combined with the areas for CNS. Now here, looking at immune response or countermeasure responses and approaches makes more sense, but you really need to nail down this non-targeted effects and these other qualitative effects, because if they don't apply in humans, then you would have a different approach for countermeasures. If they're really dominating the risk in humans, then you would target these effects for your countermeasure. So knowing what's driving the risk should lead you into what would be the right approach for a countermeasure. Thank you.